Hello friends, let us now learn some important points about oxidation of odd chain fatty acid. One of the odd chain fatty acid is propionyl-CoA. Propionyl-CoA, in the presence of propionyl-CoA carboxylase, it forms dimethyl-malonyl-CoA. This dimethyl-malonyl-CoA, in the presence of racemase, it forms L-methyl-malonyl-CoA. This L-methyl-malonyl-CoA, in the presence of methyl-malonyl-CoA mutase, it forms succinyl-CoA. This succinyl-CoA can enter into the Krebs cycle, whereas this alpha-methyl-malonyl-CoA gets converted to succinyl-CoA. Now I will put the enzymes in between. So propionyl-CoA, in the presence of the enzyme propionyl-CoA carboxylase, it forms dimethyl-malonyl-CoA, and during this process carbon dioxide is released. This is dimethyl-malonyl-CoA, and during this process, if you see, ATP gets converted to ADP and inorganic phosphate. Okay, and for this propionyl-CoA carboxylase, biotin is also a coenzyme. Now this dimethyl-malonyl-CoA, in the presence of dimethyl-malonyl-CoA racemase, it forms L-methyl-malonyl-CoA. This L-methyl-malonyl-CoA, in the presence of methyl-malonyl-CoA mutase, it forms succinyl-CoA. During this process, vitamin B12 is essential. So, just take a condition, think of a condition where there is deficiency of this vitamin B12. Then, if there is no vitamin B12, then clearly this step from alpha-methyl-malonyl-CoA to succinyl-CoA, this step does not occur. And as a result, these alpha-methyl-malonyl-CoA levels are accumulated. When this alpha-methyl-malonyl-CoA levels are accumulated, then it results in increased levels of methyl-malonic acid is increased. This is excreted in urine, and this methyl-malonic acid also gets accumulated in the CNS, leading to mental retardation, and retarded growth occurs due to this accumulation of methyl-malonyl-CoA in the CNS. So this is about the oxidation of odd chain fatty acids. So thank you guys for watching my lecture. Thank you.